Okay, thank you. Well, good morning, everyone. My name is Adriana Cero, and today I'm going to talk about my research named Response of Batwing Morphospace to Land Use Change in a Human-Modified Landscape. Well, habitat segregation due to morphological or behavioral specialization may be the result of interaction between species or as a result of environment filters that also determine the coexistence of species in the regional pool. The conversion of natural habitats to agricultural activities modifies the regional pool and contributes to the loss of species. Therefore, it is necessary to evaluate the change in community assembly through the niche of the species. First, I will specify about the Hutchinson and n-dimensional niche concept, with which a quantitative description of the biotic and abiotic conditions that enable the success of the species in a given place. Second, on the functional traits that are morphological or physiological or phenological characteristics of a species that influence her ground reproduction or survival, known as a fitness. These traits mediate a species response to the environment change that evidencing response in community assembly. Accurate example which integrates functional traits and habitat in the study was the study of Stefano and collaborators in 2020 with Cape spiders. They found that on a local scale, there is a differential specialization for K microhabitats, but there are a moderate levels of morphological similarity as we see in the figure of the right, where the colors represent the microhabitats and the morphological component the axis. We see that there is a less overlap of morphological niche and that these species have a close phylogenetic relationship as observed in the phylogeny in the middle. The framework developed here suggests that in caves, habitat specialization is mainly driven by the environment filter, and this pattern results in the establishment of communities replicate in a geographic space composed of ecologically equivalent species. Our study area was in the Serra da Bodoquena in the Brazilian Midwest. We sampled 20 communities in the dry and rainy season for these years. In field, we take wind photos and essential data for taxonomic identification. Our hypothesis was that the land use will act as an environment filter for wind shape on a regional scale. Because of this, the bat wind will be a key factor in the assembly of the bat community, as a wind shape and habitat use are closely related. So given that the environment will act as a filter, we expected to find a relationship between areas with agriculture and bats with narrow, long, and pointed wings, like insectivorous bats, in forest edge, bats with wide wings and rounded tips as animalivores. In areas with greater forest cover, bats with wide, small, and rounded wings as frugivores and nectarivores. We perform a geometric morphometric analysis. We use 2D image, always of adult males. In this, we located 18 landmarks and 30 semi-landmarks following previous studies. Subsequently, we perform a GPA analysis to obtain the pieces scores that describe the wind shape, and we perform also a phylogenetic principal component analysis. We got the first three PCs explain an 84.82% of the total of variation in the wind shape. Okay, negative values of PC1 represent narrow, low, and pointing wingtips. Positive values of PC1 represent wide, low, and rounded wingtips, and negative PC2 represent long and pointing wings, and positive PC2 represent short and rounded wingtips. The PC3 represent the less variation of the wing shape. Well, to measure landscape variables, we use Landsat satellite image and data from band biomas. To extract the variables, we construct concentric buffers of 1 and 2.5 and 5 kilometers around each sampling site. These buffer sites were chosen because they reflect the home range of most of bats and response to the habitat loss in the neotropical area. For each buffer, we calculate the number of forest fragments, forest cover, forest edge land, savanna, native forests, and crops. To reduce multicollinearity, we calculate the variance inflection factor of the landscape variables, and we keep the variables with only values less than 5, were kept for subsequent analysis. We divide the analysis into 3 parts. In the first part, we compute the hypervolume space defined by a set of independent assets, such as the trace. Here we calculate only the first 3 axes that describe wind shape and the species composition matrix with abundance data. From the hypervolumes, for each community, we calculate functional evenness and functional dispersion. In the second part, we use generalized linear models to test whether variation in evenness and functional dispersion were associated with landscape variables. And finally, we use an RLQ, which is an ordering technique among environment variables, the composition of a species, and the 3PCF of the wind shape. Here we access to the global model significance between the environment and functional traits to test the global model of 6. We found 32 species, mainly from the Phyllostomidae family. The most abundant species were Artibis planirostris, Sturnira lilium, and Carolia perspicillata. In the hypervolumen community, meta-community, we found a considerable overlap between the communities in the functional space, as we can see in the figure, where there is an overlap between the boundaries of the community that are indicated by each color line, and this suggests that the average value of the trace did not vary considerably between the communities in the Serra da Bodoquena. The evenness index was positively related to the edge, patch, crowd, and savanna, as we showed in the table, but negatively related with savanna at 5 kilometers in a bit scale. The dispersion index was positively related to the forest cover at 5 kilometers too. In the RLQ analysis, the 3 axes explain 99.52% of the relationship between trace and environment. However, the global model of the relationship was not significant. We found a negative correlation between 1 kilometer and 2.5 kilometers in agriculture, and positive values of PC2 that correspond to wide winds with rounded tips, and a positive correlation between non-phyllostomic insectivorous that have narrow, long, and pointing wings, as in Nyctilomus laticaudatus or Ectesicus furinalis. The forest edge at 1 kilometer was negative correlated with the specialist species, such as nectarivorous and animalivorous. And also at 1 kilometer in savanna was positively related to insectivorous Philostomidae as Lophostoma sylvicola or Micronycteris amborni that has broad, long, and rounded wing shapes. Also, we found a positive relationship between savanna at 5 kilometers and non-Philostomidae, and also the insectivorous and piscivorous bat that is Noctilio-Leporinus. Our results show that the land use affected the bat meta-community and filtered species based on the wing shape. But this effect was dependent on the spatial scale. On the other hand, the landscape modified by the human has effects even on in a small scale, such as 1 kilometer or 2.5 kilometers. Our results suggest that a process of functional homogenization may be underway in this meta-community. This effect may be a result of the fact that the landscape of the Serra da Bodoquena have a high demand of human land use for livestock, soy, or sugar cane, which can generate a great transformation in the landscape. Our findings are not far from the global scale patterns that show that in the neotropical region, the tendency is for mammal communities to present a high functional redundancy and a slight dispersion due to a high occupation of the functional space. It is a filter for the wind shape in these areas. The meta-community assembly is related to the availability of resource and the complexity of the habitat. And it promotes the coexistence of multiple functional groups due to their foraging strategies and the wind shapes. For example, the main reason for finding a positive relationship between agriculture and insectivorous bats is that these areas there is a great abundance of insects and arthropods. Species such as Cryptopteros or Tornatia were positively associated with savanna at fat scale, and these species have short wings with rounded tips that help them maneuver and fly slowly through dense foliage. Thus, the phytophysiognomy of the Cerrado plays a fundamental role in the presence of these species because the mosaic of this vegetation promotes this heterogeneity and increases the availability of food. Well, I want to thank to all the people who are watching this presentation, and I'm open to questions. Thank you.
All right, wonderful talk. At this point, we can now take questions. If you would like to verbally ask a question, you can use the raise hand reaction. If you would prefer to put your question in the chat, that is also fine. I guess while, people become less shy, we all get more comfortable with each other. I'm curious kind of what your next steps are. Are you going to keep working with this system and this question, or are you moving on to something else?
Yes, my idea is continue to work with morphology, but also to complement this part of my work. I'm going to work with ecolocalization, because ecolocalization and the morphology is key for the bats and his presence in determinate areas. Then I'm going to continue to reach about it because, for example, in these areas that have a high impact of for the human, for example, is key to preserve the biodiversity. Then, for example, the Parque Nacional of the Serra da Bodoquena, that is a big unit of conservation, maintain the biodiversity without care the impact of the humans, then the biodiversity of the bats is very high in this area. This is one of the main of my thesis is now find what is the impact for these bats and for the communities.
Excellent. So in this next period, we're at 15 minutes, we have a withdrawn talk. So I'm going to put the screens back up, but if anyone has any additional conversations that they would like to have with Adriana, you can put those in the chat, and we will resume at 12:30 with our second talk in the session.
Okay, thank you.
Hello, everyone. Really happy to be here. I'm Jailam. I'm a PhD student at ISEM in France, and I'm going to present some of the work I did during my master's thesis at IISER Pune, done in collaboration with Emmanuel Fraunhofer. I'm going to talk about the genetic architecture of dispersal plasticity and its consequences. And to motivate a little bit where I'm really coming from is the fact that you have a very simplified representation here of ecology on one side and evolution on the other side. And sort of for historical reasons, these fields have been separate, but that's a different discussion. But in the ecological side, you have like demographic rates and like birth and death, and also dispersal rates. But you also have the abiotic environment. And on the evolution side, you have, well, when we talk about evolution, we talk about changes in the composition of genotypes. And so we have the genotype to phenotype map. And when we think about these separately, this is how we look at them. But together, well, you have demographic rates that can impact the genotype to phenotype map, and also mutation and recombination processes that go within. And also you have the phenotype that finally feeds back onto the demographic rates. And this is where you can sort of have a mechanistic representation of ecology and evolution. And this is what we think sort of is going on under the hood. But when we as evolutionary biologists or ecologists want to study this, we develop some models. And these models sometimes can be complex to some degree and simple to some degree. And often models either have details of ecological conditions or details of genotype to phenotype maps. And so what I mean by this is, for example, you have the adaptive dynamics approaches that make really simplifying assumption on how genotypes map onto phenotypes. And making these simple assumptions like having linear relationship between genotype to phenotype and small mutation rates and effects allows us to derive, for example, optimal phenotypes under equilibrium ecological conditions, which is really great. But on the other hand, when you have complex representations of genotype to phenotype maps, often there are very simple assumptions on the fitness function, so on how the phenotype actually maps onto fitness, so which does not contain a lot of ecological details, but this allows us to give us general properties like robustness and evolvability under very abstract, like stabilizing selection or so on. And let me first talk to you a bit about the genotype to phenotype maps. So they're generally, the kind of map that I'll be focusing on is a Wagner type gene regulatory network model, in which the genotype is represented by a regulatory matrix. So these encode how genes interact with each other, and a developmental process links this to gene expression states. And the phenotype under consideration is the gene expression states, and usually you have a kind of abstract, maybe like a Gaussian function that tells you how fitness looks like corresponding to this phenotype. And what we get is often properties like robustness, where if you perturb a little bit the genotype here, you get no change in the phenotype. And this allows for evolvability, where, for example, a lot of space of genotypes can be navigated easily without a change in phenotype, and then you can have a big change in phenotype. And for the ecological part today, I will focus really heavily on dispersal evolution, and this is something that we are interested in generally. And here, for example, in models of dispersal evolution, we are always in this regime of models which don't take into account the genotype-phenotype relationship, but we have really good idea about how ecology drives evolution in this case. So let's talk about dispersal evolution for a bit. Well, dispersal is a very key process. It's a movement of individuals in space, which has consequences for gene flow. So it's a really important evolutionary mechanism, but also an ecological mechanism because it determines the fluxes of organisms in space. And often dispersal has been really well studied in terms of its ecological and evolutionary drivers, such as dispersal costs, and also the fact that spatiotemporal variability in fitness expectation and kin competition promote dispersal evolution. So this is really well understood, and often there are mathematical expressions for this as well. On the other hand, we also have dispersal evolution, which is interesting to us, not in these equilibrium metapopulation conditions, but also in the conditions of rapid change, such as range expansions. So the movement of individuals in space. And an example of this is the Australian cane toad. And what you see here is the range border position. So just how quickly organisms spread in space. And what you can see is that dispersal evolves under these conditions by a process called spatial sorting, in which faster dispersal genotypes end up at the range expansion front. And this really leads to accelerating range expansion dynamics. And this was shown by Perkins et al. In 2013 in the cane toad example. And so moving on to models of dispersal evolution, as I said, dispersal evolution is generally modeled over here. So you assume very simple genotype to phenotype maps. This was reviewed by Sastamoinen et al. And these simple genotype to phenotype maps under equilibrium conditions give us optimal dispersal rates and plastic responses. So by plastic responses, I mean that often dispersal need not just be a fixed rate, but it can respond to local conditions, such as population density. And this leads to reaction norms rather than just particular rates. And we can just derive them sometimes even analytically and many times in simulation using these kind of approaches. And today I'm going to focus on density-dependent dispersal. Density-dependent dispersal, what we mean by this is that dispersal probability is a function of the local population density. And how this really works, we know now, going to the ecological conditions, the details of the ecological conditions, what we know is, well, if you don't have any kind of negative relationship between local population growth rate and population density, you would have exponential growth. So often this is the case. So if within a patch in a metapopulation, you have lower population densities, so on an average growth rate is greater than expected, then individuals do not disperse. And then if it's lower, they disperse with a saturating probability. And this was derived mathematically by Putka and Hoerstadt using a marginal value approach. And of course, these kind of approaches use very simple assumptions on genotype-phenotype map, assuming that the optima can just be reached. But under these rapidly changing conditions like range expansion, we are not just interested in evolutionary optima, but how fast those optima are attained. So for this, we also want to include some details of the genotype to phenotype map. And so here we develop an individual-based model of a gene regulatory network model for density-dependent dispersal. So I'm not going to go into too much detail, but I'll give you the information that you need to understand the results. Well, you have a discrete time, discrete space model with organisms with non-overlapping generation. All kind of density regulation is local. And we have a gene regulatory network that encodes dispersal. So you have these regulatory genes, like in the Wagner model, that can interact with each other, but they also respond to the external conditions, that is the population density within a patch, and they produce as an output dispersal probability. And we know now very well what the theoretical expectation is in this kind of metapopulation, so we can actually optimize the threshold of this function. And so what we want to know first, we allow this to evolve under equilibrium metapopulation conditions for 20,000 generations. And then we allow for range expansions to begin. And then we are interested whether these two approaches, so a standard approach where we just optimize the threshold of this function versus a gene regulatory network model approach, actually impacts range expansion dynamics. And just few more model assumptions to see here is that dispersal is local and it is costly. So individuals die with a particular dispersal probability, and there are random patch extinctions at each time step. So patches may go extinct due to some kind of a disturbance event. And this generally promotes dispersal evolution as a bet hedging strategy, but we also see some interesting things emerging from these kind of conditions. And so we first ask the question, does a gene regulatory network model lead to emergence of the theoretically expected plastic response in the equilibrium metapopulation condition? And in order to answer this question, well, here are the results. You have dispersal probability on the y-axis, and on the x-axis, you have population density. For From left to right, dispersal costs increase, so you have higher dispersal mortality, and from top to down, the patch extinction probability increases. And what we see here is that at low dispersal mortality and low extinction risk, this is the reaction norm obtained. And here we only show those conditions that occur during the in under equilibrium metapopulation conditions, because selection only can optimize the reaction norms over there. And we see that indeed, the gene regulatory network models produce theoretically expected reaction norm in conditions of population densities that occur in equilibrium metapopulations. And moving on, we can next ask this question that, okay, now that we know that these models are sort of equivalent under equilibrium metapopulation conditions, at least under those conditions where, under those population densities that occur, what are the impact of range expansion dynamics here? And to see this, we have on the y-axis again, the range border position as a function of time. And what we see here is that at low dispersal costs and low extinction probability, you have faster range expansions in the gene regulatory network model compared to the standard approach of optimizing the threshold. And the reason for this is because you have in equilibrium metapopulation conditions, only optimization happening at the densities that occur. So this region here, which is not optimized during equilibrium conditions, actually can maintain what we are known as hidden genetic variation, which is released during range expansion, because these conditions are experienced of lower densities are experienced during range expansions. Further, we have here at high extinction probabilities, where you actually have a wider range of conditions, ecological conditions occurring. And so you see that at low population densities as well, you have a good match between the two approaches of optimizing the threshold and the gene regulatory network model. So you have very similar dynamics in range expansion. So it's actually faster range expansions happen in the gene regulatory network model due to maintenance of variation in conditions that do not occur under equilibrium metapopulation. And this, in summary, gives us a take-home message that we actually have now a model where we have the details of the genotype to phenotype map and the details of ecological conditions. And so when we go from here and include details of the genotype to phenotype map into these models with realistic ecological conditions, we say that we have an emergence of, so they're equivalent under equilibrium conditions where ecology and evolution happen on the same time scale. But we see diverging dynamics, diverging ecological dynamics when ecology and evolution don't happen on timescales, for example, in range expansions. And the second interesting, I think, perspective to look at this is that when we actually include ecological details in genotype to phenotype maps, what we get is that we have hidden variation as a function of ecological parameters rather than some kind of an abstract selection function. And for this, I'd like to acknowledge my master's thesis advisor and my current PhD advisor, Emanuel Fraunhofer, and the INF for funding this research. And I will also be presenting in person, so I'll be talking a little bit more on genetic architecture and GRNs in a poster, and also generally on my PhD work on eco-evolutionary dynamics and host parasite systems in complex landscapes in a talk. Thank you very much.
Excellent talk, very exciting work. At this time, if anybody has any questions, you can use the raise hand mechanism and I'll invite you to unmute it. Otherwise, it looks like we already have a question there in the chat for you. It says, can we get an example of regulatory genes that work during population-dependent dispersal?
Yes, so yes. So I think this has not really been looked at very explicitly in terms of regulatory genes there, but there have been, for example, studies that look at systems like P aphids, which in which you can have the proportion of winged and unwinged morphs that are produced. So this is like a very clear developmental mechanism. And often like the proportion is induced by crowding or by density, if I'm not mistaken. I'm not an empiricist, but this is how I understand this. And so you can have regulatory mechanisms which kind of via development lead to different dispersal rates. So yes, I think the empirical work here is in there. This is a bit out there, trying to provide a mechanism. as well. Like, so this is also an important thing where we are trying to really provide a mechanism for what we have kind of theoretically in a very abstract way. So yes, so there's some work there.
Wonderful. Are there any further questions for our speaker? All right. Well, thank you very much. I think we're about time to start moving on to our next talk. So thank you very much. That was very, very cool. All right. And our next speaker, Samuel Greaves. I know you are here. If you would like to get your screen share up, you are welcome to do that at this time. Oh, it looks lovely. I'll let you know when to start.
Okay.
All right, you are free to go whenever you'd like.
Okay. Hi, I am Samuel Greaves, a PhD candidate in the Gaither Lab at the University of Central Florida. And today I'm going to talk about the color polymorphism of the archaea hawkfish Paraserites archaeus. So color is an important part of life, particularly on reefs. It can be used to hide from predators and prey through camouflage and mimicry and affect survival, or send conspicuous signals for things like mating. Color polymorphism is when there are multiple discrete color patterns within a species at rates higher than can be explained by drift, and it is commonly linked with sex ontogeny. Color polymorphism is common in fish, but so are strikingly different colorations between sister taxa. Because appearance is tightly intertwined with mate choice, such as through mate recognition and sexual selection, as well as survival, color and color polymorphism can be involved with speciation. I'm interested in researching non-sex-linked stable polymorphisms and will study this using hawkfishes. So Paraserites is a genus of hawkfish containing 6 species distributed through the tropical Pacific and Indian Oceans. At least 3 of the species here on the top row display color polymorphisms across their ranges. My dissertation work focuses on Paraserites archaeus and Hemistichtis. These 2 species have a similar light melanistic dimorphism, where the light morphs are characterized by a white stripe and a countershaded color pattern, while the melanistic morphs have a dark coloration throughout their bodies. There are also some intermediate phenotypes. And this talk will primarily look at my preliminary results from archaeus. It is important to know that hawkfishes have a harem-based mating system where a larger male, here in the red circle, will defend a territory encompassing the territories of 1 or more females. For archaeus, they are generally centered on a cluster of coral heads and are only a few meters wide. This territoriality allows us to look at the distribution on a relatively fine scale. This study was inspired by some work done by Dr. Jonathan Whitney, where he set out to identify whether the color morphs of Paraserites archaeus in Hawaii were diverging. Using transect data, they were able to associate color morph with niches constructed from environmental variables. Melanistic morphs were more frequent right below the reef crest in a shallow, steep, high-energy environment where Populipora corals dominate a low coral cover portion of the reef. Meanwhile, the light morphs were more likely below the surge zones, where calmer water results in a high coral cover, primarily Paraites species. They also conducted genetic analyses using 30 microsatellite markers. Using the full set of markers in a structure analysis, the color morphs were indistinguishable. However, with a subset of 7 loci, they were able to show signals of differentiation. Additionally, the intermediate phenotypes showed the approximately 50-50 split we would expect from F1 hybrids of the 2 morphs. Graphs plotting FST against heterozygosity are often used to find FST outlier loci. So in addition to the previous microsatellite markers, they sequenced the melanocortin-1 receptor gene, or MC1R, and plotted them here, showing that a SNP from the MC1R gene is significantly differentiated between the morphs. This is important because this receptor is known to affect the level of the dark pigment melanin in other species. Whitney et al. Suggested that the shift in proportions by morph in both allele frequencies and habitat usage indicate that the color morphs were in the early stages of divergence, and that their color is potentially linked with crypsis. On reefs, shallower waters are counterintuitively kind of darker due to turbidity and a higher amount of algae-covered rock, and shadows in the rocks and branching corals when backlit by the sun. In these unedited photos on the right, we can see that the melanistic morph blends in with the shadows cast by this coral head. Meanwhile, the pink color of the light morph appears green at the depths that they are found in, matching the ambient colors of the reef and blending with the corals themselves, which have broader lobes than the shallower varieties. Hawkfish hunt by perching on these coral heads, then swooping down to strike prey. So staying hidden from prey may improve their success at predation. My goal is to gather more data about these hawkfish color morphs to see whether this pattern is broadly applicable and hopefully tie it to function. I first conducted a geographic replicate of Whitney et al.'s ecological study, but in Guam, to see whether their preferences extended to other populations. Guam is less isolated from the core of the species range in the tropical West Pacific, and the reef ecosystem there is much more diverse. I also conducted whole genome resequencing to try to characterize any genomic regions that have ties to color morph. These will be useful to identify the mode of evolution of this trait and other traits that may be linked with it. And then finally, my last objective is including a second species for both of these to gain additional insight into the age and stability of the polymorphism. I obtained samples for genetic analysis for both species from Guam, and then Dr. Whitney provided me with Arcata samples from Hawaii. I also did some observational work, which was conducted across several sites along the west coast of Guam. In total, we surveyed 5,300 square meters of reef using 25 by 4 meter transects, finding 455 observations of Arcata, including 267 light, 153 intermediate, and 32 melanistic observations. Unfortunately, with only 21 hemistictus, I did not obtain an analyzable sample size, so today I only present my analyses on Arcata. So starting off, the morph frequencies for Arcata were very different from the Hawaii study. There, the morphs were almost evenly split at around 40% each, but the light morph dominated my Guam transects, and the melanistic morph was only 7% of my observations. Additionally, the overall density was far lower, about 30% of Hawaii's, giving us an indication something different might be happening on Guam. To look at whether the environmental variables I collected, which are up on the right, were able to explain the morph proportions of the transect, I ran redundancy analysis. I started off with the final model from Whitney et al., which in the Hawaiian system was able to explain 64% of the variation in transect morph proportions. However, on my Guam data, the model performed poorly, explaining only 8% of the variation and was not significant. I then used forward selection to build a new model, which resulted in using only the percent cover of rock and Populopora species. This model performed over 4 times better at 36% and was significant. In this case, the melanistic individuals are more likely to be found where there is a higher Populopora cover and less bare rock. Next, I examined the spatial distribution of the morphs. Looking purely at depth across all observations on Guam, since that was a major explanatory variable in the Hawaii study, there is a slight drop in median depth of the light morph, but overall there is high overlap in the depth distributions by morph. Next, looking at the transect level, I calculated Levin's overlap index. This value will be 1 when the pair co-occur and are equally abundant per transect, and 0 when they never co-occur. And 95% confidence intervals are below the observed value. The melanistic and light morphs index is about 0.4, which does not indicate a perfect overlap or separation, but instead a kind of uneven overlap. So now that we have evidence that the color morphs are overlapping in depth and on transects to some extent, another opportunity for segregation is in harem formation. So this graph shows the data at the individual level, looking at each observation's nearest neighbors, which are the morphs within 4 meters. Each bar represents an individual and the morphs within its radius, with the lowest rectangle being the focal individual. Again, hawkfish are territorial, and this radius likely captures a harem. Note that the data is shown for only 3 of the 5 sites because we did not observe any melanistic individuals at the Barracuda and Turtle Rock sites. So for example, these 4 individuals comprise each other's neighbors, and the furthest right one is also in range of the next fish down the transect. There's only one single cluster that is majority melanistic. However, there are many examples that you can see of mixed potential groupings, indicating that there is not a hard barrier in mating opportunity between the morphs. So overall, these survey data show that there is a higher degree of overlap between morphs on Guam than was observed in Hawaii. So moving on to the genomic data, my lab has produced a high quality reference genome for Arcadis, about 690 megabases with 24 chromosome scale scaffold. The BUSCO score is about 98%, and the annotation contains almost 19,000 genes. To leverage our genomes, we conducted a whole genome resequencing on 228 samples, of which 93 were Arcadis from Guam, and 71 were Arcadis samples from Hawaii. I also sequenced several other species as outgroups for some of my later analyses. Using PCR-free library prep to get a more even coverage and minimize error, we got almost 14 billion paired end reads from Illumina NovaSeq sequencing. And after quality filtering alignment to the genomes and SNP calls, I ended up with tens of millions of SNPs for these different groups. I've not gotten very far in the genomic analysis yet, but I did conduct some FST tests using 5 kilobase sliding windows long genomes. So I did these by morph within a location, which are the top 2 graphs, and then between Hawaii and Guam, which is the bottom graph. As expected, there is greater divergence between the by geographic locations than by morph. So note that the bottom scale is about 4 times the top. However, there are 2 important things. First, Hawaii has a higher magnitude and a greater number of outliers than Guam. And then second, there are outliers that are different between the groups. I've placed orange boxes around positions that are outliers when FST is calculated by morph, but not by the 2 islands, and then blue boxes around the opposite. So zooming in on these, there are outliers by morph in those that are outliers by morph in both locations, but are not outliers between the islands, are strong candidates for being under selection and linked with color phenotype. So to sum things up, while my analysis is hindered by some low sample sizes of melanistic individuals, what is clear is that there is less pronounced niche segregation in Guam driven by different factors. But I did recover the greater genetic divergence in Hawaii, meaning that the Hawaii population may be following a more diverging evolutionary trajectory between the morphs. My next steps are to do an additional field season after modifying my transect protocol to get sufficient observation coverage for melanistic individuals and those hemistinctus second species. And then for both Arcadis and hemistinctus, I will conduct additional genome scans and selection tests to identify regions under balancing or directional selection or have high linkage disequilibrium. Then I'll also run genotype association with the color morphs to identify candidate genes that are associated with the morph, to learn more about the genetic architecture and history of the color morphs. And with that, I'd like to acknowledge my co-authors, funders, and everyone involved, particularly the undergraduates that helped me process a lot of photos, and the techs at the University of Guam Marine Lab who helped me with my collections and fieldwork. And thank you all for listening, and I am open to questions.
Excellent talk. At this time, we can now accept questions. If you would like to ask a question, you are welcome to raise your hand and I will request that you unmute. If you are not interested in asking a question verbally, you can also put one in the chat. I see we already have one in the chat and I'll read that so that it's on the video for future viewers. All right, a lot of data. Where do the 2 morphs reproduce?
So they are broadcast spawners. So they reproduce in the upper portion of the water column, but they do this in like those harems. So they have like a small, they're quite restricted in those pairs in terms, they don't form mating aggregations or migrate to particular areas for reproduction. They pretty much stay in the same spot once they're adults and reproduce there. Yes, there is overlap in the range of the two morphs completely. They're found, both morphs are found across the entire range of these species, and both morphs are found on the same reefs and even the same coral head sometimes across that entire range.
Just for completeness of the record, for when we're viewing this later, the question was, is there a range overlap in the range of the two morphs?